Hi everyone, and welcome back to Biology 206. I'm Dr. Jennifer Belzer, and today we're going to be talking about energy, how organisms make a living. Now we know lots of organisms consume food in order to produce the energy that they need, or in order to gain the energy that they need for their various energetic requirements in their bodies. But we're also going to be talking, today's emphasis will be on autotrophs. So those organisms that are able to produce energetic compounds, produce carbon-rich compounds using carbon dioxide, as opposed to other sources of carbon. First, we're going to talk about chemoautotrophs, or those autotrophs that use energy tied up in various chemicals in order to help to fix carbon dioxide into sugars. And so instead of, we're all very familiar with photosynthesis and the use of light as an energy source to fix carbon dioxide. What chemoautotrophs do is generate energy from other chemicals. So in this case, these sulfur bacteria use hydrogen sulfide and the bonds in hydrogen sulfide to help convert carbon dioxide to sugars, leaving behind the residue of elemental sulfur. And so this is what we see happening in this particular diagram. A lot of these chemoautotrophs live in places where there is no light, and that's where the evolution of many of these organisms occurred. And so it makes a lot of sense to have to make use of other sources of energy to drive this autotrophic lifestyle. So some other ways that organisms can fix carbon, other places where they receive this electron donation. So again, remember, photosynthesis is driven by light, and that excites electrons, which become the electron donors for that carbon fixation. In contrast, in chemoautotrophs, we use various other compounds that donate electrons to help with that carbon fixation process. And these drive all kinds of different functions in ecosystems around the planet. Okay, to kind of get into this, I wanted to show this really quick video. We won't go through all of it. I'd encourage you to watch this yourself, but it kind of demonstrates, it talks about the evolution of these chemoautotrophs and the amazing contributions they make to productivity of various extreme environments. So a lot of these chemoautotrophs are what we call extremophiles, and they help support the communities living in those extreme environments. So I'm going to play a little bit of this video. Please finish watching it on your own, but this will give you a sense of what these places look like.
The majority of life on the planet is based in a food chain which revolves around sunlight. But of course, in the deep sea, there is no light. Once you go below 200 meters, light just disappears. That means there are no plants down in the deep ocean. So there is a food chain that operates completely independently. Instead of having sunlight being used as the primary form of energy, chemical energy is used. And that is from these chemical-rich fluids such as methane and also hydrogen sulfide that leak out of the seafloor from fissures that have been formed by tectonic activity. And instead of there being plants, there are bacteria which act as the primary producers, and they utilize this chemical energy in a process known as chemosynthesis rather than photosynthesis, which is pretty amazing. So in geologically active areas such as the Gulf of Mexico, the hydrocarbon-rich fluid and gas can result in a number of unique and rare environments that can range from, a small patch of blackened reduced sediment to cold seeps with extensive mussel beds. Once you see a cold seep, it's just booming with animals. There are thousands upon thousands of animals in some cases. And that's because they've got a really plentiful energy source. So those hydrocarbons seep out of the seafloor, the bacteria then use them, and that bacteria is just constantly being replaced once it's eaten.
Okay, so I just wanted to give you a little tidbit of that video to kind of show you how much amazing life is supported by these primary producers that we don't think about very often, these chemosynthetic organisms. So please do continue watching this video. It walks through all kinds of amazing things that chemosynthetic organisms do in these deep sea environments. And feel free to explore other places where we have chemosynthesis happening, and they again tend to be these really harsh, extreme environments. Okay, but of course, for most of the planet, it is photosynthesis that fuels life. So this is a satellite-based image of that shows carbon absorption on the land and in the oceans, based on spectral reflectance measured from satellites. And so you can see how much activity there is in terms of carbon absorption, and also places that don't have any. So here are those deserts we learned about a couple of weeks back, for example. Okay, so we're going to be moving on and talking a fair amount about photosynthesis and different ways that photosynthetic organisms have to deal with environmental stressors. Okay, so one thing I want you to keep in mind is the fact that the evolution of photosynthesis really changed the trajectory of life on Earth. So there was a very long period of time where there were no photosynthetic organisms. There certainly would have been those chemosynthetic organisms we just learned about, which are much, much older than photosynthetic organisms evolutionarily. But there was a long period of time where there was no photosynthesis. And so the interesting thing that you can look at in the context of that is the fact that there was also no or very little atmospheric oxygen. We know how important oxygen is to us and many, many other organisms. So until the evolution of photosynthesis, there was very little oxygen in our atmosphere. So our atmosphere was incredibly different compared to what we know today. And so this red line depicts the increase in, or a hypothesized scenario of what the evolution of atmospheric oxygen looked like on the for atmosphere and surface ocean oxygen concentrations look like. And what we can see is that once we had the evolution of photosynthesis, we see this increase in oxygen concentrations. It took a while for the oceans to reach capacity in terms of oxygen concentration, and then for the lands to reach capacity in terms of oxygen concentrations. So it took quite a while for atmospheric oxygen concentrations to respond to the evolution of photosynthesis. But what we see is that connected with the evolution of oxygen in our atmosphere, the evolution of photosynthetic organisms that produce oxygen as a byproduct, was the evolution of a whole diversity of animals. So that happened fairly recently. So vascular plants, so plants that were able to live on land, vascular plants that have a water transport capacity, and the evolution of animals happened around the same time once oxygen concentrations reached a sufficient level in the atmosphere. Or this is one of the hypothesized mechanisms supporting the diversification of those groups of taxa. So evolution of photosynthesis that happened a really, really long time ago, way back here, between 3.8 and 2.5 billion years ago, was a really pivotal moment on Earth. Okay, so photosynthesis, very important. Now, of course, this is just a little review. You would have learned about this last year in introductory biology, but just a little review of what the photosynthetic processes look like. We have photosynthesis. Unlike those chemosynthetic organisms, it is fueled by the energy by photons of light that come and excite electrons as part of the light reactions. So photosystem 1, photosystem 2, and electrons are excited through those photosystems, and then that energy helps to phosphorylate ADP and add hydrogen to NADP+, those energy sources fuel what we refer to as the Calvin cycle or the Calvin-Benson cycle, or the dark reactions of photosynthesis, during which carbon dioxide is fixed and sugars are produced. This is a very, very simplified form of this. It's certainly worth investigating those processes in more detail, but that's I just wanted to do a little review of that photosynthetic, pathways that lead to sugar production, the fixation of carbon dioxide and sugar production, which really is what fuels that carbon signature we saw in the satellite image from a few slides ago. Okay, so when we're thinking about the kind of organism we have, the key distinction between autotrophs and heterotrophs is that autotrophs synthesize organic compounds from CO2 plus an energy source. So we can think about our photosynthetic organisms that use light as an energy source and CO2 to produce sugars and oxygen. Okay, that oxygen that led to big changes in our atmosphere. And then chemosynthetic organisms, in this case, we're looking at those sulfur bacteria, but we know that there are different chemicals that can be substituted here. So carbon dioxide plus the energy from a chemical, in this case hydrogen sulfide, plus oxygen leads to the production of sugar, plus sulfur, plus water. So the take home is that both of these types of autotrophs can use carbon dioxide and convert that carbon dioxide to sugar in the presence of an energy source. There are two others. So when we think about this, we can start obtain carbon elsewhere. So do these organisms, can they use carbon from the atmosphere, or the carbon in the water, or carbon in the atmosphere to produce sugars? And if the answer, if they can do that, then they don't need to obtain carbon elsewhere, and you go over here to our autotrophs. If they do need to obtain carbon elsewhere, you go over here and you can look at whether they use energy from light to contribute to this process or not. And so we fall in the not category. So we need to obtain our carbon from elsewhere. We cannot use light energy to help support that process. So we as people and many, many other organisms on the planet are chemoheterotrophs. Okay, so we're heterotrophic. Photoheterotrophs, in contrast, are able to use the energy from light to help support the production of carbon compounds for use in their daily energetic needs, but this is done through a two-step process. So they have to break down a carbohydrate to produce malate and CO2 and energy, and that CO2 is then used in combination with another carbon compound, pyruvate, plus ATP, that phosphorylation happens thanks to photons of light to produce a carbon compound, and then reduce this to ADP. Okay, so that's the difference. So there are some organisms that can use light but are not able to use carbon dioxide for the production of those carbon compounds. Those are photoheterotrophs in comparison to photoautotrophs, which use atmospheric carbon dioxide to produce the carbohydrate, the forms of carbohydrate that they store and use for energy sources. Okay. It's not totally as clean cut as that. We know that not all plants are fully autotrophic. Here we have a couple of examples. One is this insectivorous plant or carnivorous plant. In this case, it's a Venus flytrap, but we know there's lots of plants that have evolved the ability to trap and digest insects because they live in very nutrient-poor environments that don't support sufficient carbon dioxide uptake through photosynthesis. Okay, so other forms of nutrition and energy supply have been developed through this process, through the evolution of carnivorian plants. And then the other one is parasitism. So there are lots of plants that have just mostly dropped the ability to photosynthesize, or entirely dropped the ability to photosynthesize, and rely entirely on resources from other from other plants. And so this is a Cuscuta, or a daughter. Dr. Mihai Costia in my department, and our department studies these extensively. And this is a parasitic plant that actually, takes resources from the phloem of its host plant, and also uses its host plant for support. But so it does not produce, even though it's a plant and has probably the ability for some photosynthesis, we see some greenness there, most of its nutrition, most of its energy requirements are provided by a host plant. So there are some different ways that plants can earn a living. And likewise, not all animals are fully heterotrophic. So this beautiful little image here from our first slide is a sea slug. This is Alicia, and they're absolutely gorgeous. What happens is that they ingest chloroplasts from different when they're feeding on plants in the ocean. And they incorporate they consume some of those chloroplasts, but some of them are incorporated into specialized structures that give them this green appearance. And in fact, some of their nutrition is derived from the ongoing photosynthesis of those chloroplasts. When you put these slugs into the dark, they don't grow as well as when they're in the light. Okay, so this is referred to as kleptoplasty, which is a pretty fun term. So stealing the plastids from something else. And we know that that's something that has happened in various evolutionary changes. Okay, so this is the digestive tubule of a sea slug, Alicia clarkii, packed with chloroplasts taken from this algae that we can see this sea slug feeding on. So the C's are chloroplasts, and the N is the cell nucleus of that digestive tubule. So pretty neat. So we have some animals that are able to actually use photoautotrophic mechanisms to support some of their nutrition, not all of it, of course. Okay, so now that we've gone through some of those little oddities of our typical breakdowns, what we're going to look at is how photosynthesis is affected by environmental conditions and some of the amazing evolutionary strategies that have been evolved to cope with environmental stress and maintain photosynthetic capacity. Okay, so first, as hinted at in the previous slide, is that light availability is really key. Of course, photosynthesis relies on light to excite electrons and drive that production of ATP and NADPH to feed the Calvin-Benson cycle. And so if you are, this is what we refer to as a light response curve for photosynthesis. Here on the y-axis, we have photosynthetic CO2 uptake rate. And this can be in various units. You can think about it as a flux of carbon dioxide per unit area per second, for example. So micromoles of carbon dioxide per meter squared per second, which is what we have over here in this figure. And so when we're in the dark, so zero is dark, zero is no photosynthetic rate, sorry, and zero on the y-axis, on the x-axis is the dark. So we have on the x-axis a scale from dark to full sun, which is about 1,500 micromoles of photons per meter squared per second. Okay, so when we're in the dark, no photosynthesis is happening, and in fact there's a negative photosynthetic value, and that's because of respiration. So those metabolic processes that happen in the cell just to support sort of all of the costly things that a cell has, or that a leaf has in it. So rubisco and different other enzymes and proteins that support cellular function. So all organisms have these respiratory processes. And so when you put a leaf in the dark, its photosynthetic rates are negative because it's actually producing carbon dioxide through respiration. So it's consuming oxygen, it's using those oxidative pathways we talked about in the last lecture, consuming oxygen to break down sugars and produce energy for cellular function, and that produces carbon dioxide, of course. And so carbon dioxide is released when the leaf is, well, it's released all the time, but it's most noticeable when your leaf is in darkness. And as the light increases, that photosynthetic rate increases. And so until we reach this maximum point of light saturation, that leaf or that individual is experiencing light limitation. So its photosynthetic rate is depressed compared to where it would be if it was in full sunlight conditions for that plant. And different species have different full sunlight conditions. You can see that here for plants grown in low, medium, and high light, that the maximum amount of sunlight required for a leaf of a plant grown in low light is lower than the maximum amount of sunlight required for a leaf grown in high light. And so that light saturation varies from individual to individual depending on light environments, and from species to species depending on their life history, in terms of really productive or very very light demanding or very shade tolerant species. Okay. There are these plastic responses in the leaf too. So we just looked at these plastic responses of these photosynthetic light response curves. Oh, and I should mention that where this curve crosses zero, so the place where respiration is balanced by photosynthesis, is referred to as the light compensation point. And that's really important because it tells you the light level at which you kind of balance, you're producing enough carbon to balance your carbon losses. Okay, so we looked at these plastic responses of this light response curve for, in this case, it looks like some kind of nettle grown at 3 different light environments. And we see these plastic responses of the physiology with increasing photosynthetic rate and also increasing respiration rate for plants grown under high light conditions. Okay, so they respond plastically. And this is what the plastic responses in the leaves look like. So leaves grown of a single species, leaves grown in full sun, are thicker than leaves grown in shade. And it's a really interesting distinction between that and adaptive responses to light, because shade tolerant species tend to have thicker leaves compared to light demanding species, because those shade tolerant species have to invest in longer lived tissues where they are avoiding being damaged, etc. So these are the plastic responses we see if you have individuals within a species growing across light environments. Now, these plastic or acclimatory responses differ from adaptive responses to light availability. So this is actually work that I did during my PhD when I was, maybe just a little bit older than you folks. And here we measured shade tolerance, so whole plant light compensation point. So that point where, if you recall the light compensation point, it was where photosynthesis balanced respiration. In this case, whole plant light compensation point is where your carbon gains at the whole plant level meet your carbon losses at the whole plant level. And what we see is this, we have a whole lot of different variables here. These are all part of sort of a leaf economic spectrum. We have photosynthetic rates here. A is the depiction of photosynthetic rates, respiration rates, tissue nitrogen, so leaf nitrogen, leaf mass per area, how thick that leaf is. A higher leaf mass per area is a thicker, denser leaf. And leaf lifespan, so how long does the plant hold on to its leaves? And what we see is that more light demanding species, so plants that have higher whole plant light compensation points, also have faster physiology, so higher respiration rates, higher photosynthetic rates, thinner leaves, shorter leaf lifespans, and higher tissue nitrogen concentration. So there's these suites of traits that go hand in hand with being light demanding versus shade tolerant that have to do with producing durable, tough tissue where light is limiting. Okay, so this is a plant economics, the fact that plant economic trade-offs dictate the set of traits that are possible. So you don't ever have plants that have a low whole plant light compensation point or are very shade tolerant but have incredibly high nitrogen concentrations or incredibly high respiration rates. Those are sets of traits that cannot go hand in hand. They are not sort of stable sets of traits from a growth and productivity perspective. Okay, so in addition to light stress, temperature is a really major limitation on it really has a major impact on photosynthetic rates in plants. And so here we see, this is very busy because I've used the image from your text that has all the notes, but here we have tissue temperature and photosynthetic rates. So again, these are referred to as temperature response curves. And so you measure photosynthetic rates at different temperatures and you look at how the physiology of that species responds. And so we can see, first of all, very big differences in peaks. And so for this species here, Neuropogon, an Arctic lichen, it can photosynthesize at freezing temperatures, and its sort of optimal temperature is here around 5 degrees Celsius. Ambrosia, it's a shrub that grows in cool coastal environments, and its peak photosynthetic rates are around maybe 15 degrees Celsius. Then we move over to Atroplex, which is an evergreen shrub in deserts, and its optimal functioning photosynthetic optimal temperature for photosynthetic function is about 30 degrees. And then we have Tidosdromia, which is a desert herb, and it can function at photosynthetic rates that would denature those proteins and enzymes in many plant taxa. So we have this incredible diversity of responses to temperatures that are a function of the environment that that plant has to grow in. But we can also see ecotypic divergence. So here we have this same species, Atroplex, growing in a desert population versus a coastal population. When we grow both of those populations at 23 degrees Celsius in day, 18 degrees Celsius at night, they show similar response curves with a peak temperature of about 30 degrees, which is what we see up here. When we grow them at high temperatures, the desert population is able to respond and maintain photosynthetic rates despite high growth temperatures, whereas the photosynthetic rates of the coastal population, which would experience cooler temperatures, it's simply not able to respond, and we see reductions in photosynthetic rate in response to temperature. So temperature is a really big stressor for many plants, and this can show up as adaptive differences and ecotypic differences, and of course plastic differences. Okay, so we have these temperature issues, and then there's another problem is that ribulose 1,5-bisphosphate carboxylase oxygenase, also conveniently referred to as Rubisco, it's a really kind of a sloppy enzyme. So for those of you who are familiar with enzyme terminology, carboxylase/oxygenase means that Rubisco can fix carbon dioxide, but also oxygen. And so it has a higher affinity for carbon dioxide, but this enzyme evolved in an environment when, so if you recall, back to this diagram, photosynthesis evolved, so ribulose bisphosphate evolved, or Rubisco evolved during a time when atmospheric oxygen concentrations were very low. And so this would not have been a problem because atmospheric carbon dioxide concentrations would have been very high. But as that oxygen concentration in the atmosphere increased, all of a sudden there's a lot of oxygen to compete for that binding site on Rubisco. And so we have what is referred to as photooxidation, where oxygen is taken up instead of carbon dioxide. And this happens when this happens, it is very costly to the plant. So the photooxidative process does eventually lead to sugar production, but it reduces the efficiency of those reactions by about 25%. And so about 25% of reactions by Rubisco and C3 plants lead to oxygenation, not carboxylation. But this also leads to between a 25 and 40% reduction in energy, a 25 to 40% increase in energetic waste when oxygenation takes place as opposed to carboxylation. Okay. So the product from photorespiration cannot be used by the Calvin-Benson cycle, and so it has to go through a whole other series of steps to bring it back to products that can be used in the Calvin-Benson cycle. And so this is where a lot of that energy loss happens. Okay, so we have this sloppy enzyme that is used, that is the key enzyme used in carbon dioxide fixation for photosynthesis. And when you have hot temperatures, we end up with a lot more photooxidative stress, so a lot more photooxidation happening compared to when temperatures are cool, and also when carbon dioxide concentrations are high versus when they're low. We have a lot more photooxidation when the relative proportions of carbon dioxide to oxygen are low compared to when they're high. So plants, not surprisingly, have evolved some pretty cool adaptations that are seen really commonly in hot and dry environments. So the first of these is C4 photosynthesis. So when we talk about C3 photosynthesis, we're just talking about that, that very typical light and dark reactions. You produce, you excite electrons, you phosphorylate ATP, you produce NADPH, and that's passed to the Calvin-Benson cycle directly. And so when we think about these adaptations, what they're really focusing on is removing that photooxidative that opportunity for photooxidation. And so the way they do that is by concentrating CO2 around the places where carbon dioxide fixation occurs. So where we have rubisco. Okay, so, and the way this happens is by specialized structures, which we refer to as bundle sheath cells. And so when you slice open a leaf, when you take a section of a leaf, you can tell very clearly whether it's a C4 or a C3 plant. C4 plants have these bundles of sheath cells around the xylem and the phloem. These bundle sheath cells around the xylem and the phloem. So what happens is you have carbon dioxide that comes in through the stomates. That carbon dioxide is fixed by PEP carboxylase. So phosphenyl pyruvate carboxylase. And this enzyme is specific to carbon dioxide, so it avoids making those mistakes of that rubisco makes in sometimes taking up oxygen, sometimes taking up carbon dioxide. So what happens is bicarbonate is added to phosphenyl pyruvate to produce a 4-carbon compound, oxaloacetate, which is passed to which is then broken down here in the bundle sheath cells. So it's passed to the bundle sheath cells. Oxaloacetate is broken down into a C3 compound again, into phosphenyl pyruvate again, and CO2 is released into these bundle sheath cells. So to just walk through that again, we have CO2 taken up into the spongy mesophyll, where PEP carboxylase adds that CO2 onto phosphenyl pyruvate to produce oxaloacetate. That C4 compound, oxaloacetate, is passed to the bundle sheath cells, where it is broken into carbon dioxide and phosphenyl pyruvate. The phosphenyl pyruvate cycles back to the spongy mesophyll cells, and the CO2 is put into the bundle sheath cells. This means that the concentration of CO2 in these bundle sheath cells is high and minimizes the opportunity for oxygen to beat carbon dioxide to the punch in the activity of rubisco. And so rubisco becomes much more efficient in terms of fixing carbon dioxide compared to in a C3 plant. So we have this spatial separation of CO2 uptake and CO2 fixation that helps, and a carbon dioxide concentrating mechanism that helps to avoid photorespiration. Super cool. Okay, and there's the little equation of for this process. Okay, and the neat thing that we can look at is the relationship between the prevalence of C4 plants, so in this case C4 species in regional grass floras, as a function of average January minimum temperature. And we see this really, really strong relationship. The hotter it gets, the higher the proportion of species in that particular community will use C4 photosynthesis to avoid that photooxidative stress. Okay. This pathway also really greatly increases water use efficiency, because even when stomates are closed, because you've concentrated carbon dioxide in these bundle sheath cells, so stomates can be closed because you have this spatial separation of CO2 uptake and CO2 fixation. You can close your stomates and continue with photosynthesis. This is not the case in C3 plants. Once you close your stomates, you start to reduce your photosynthetic efficiency, okay? And so C4 plants can be much more water use efficient, which is really handy in these hot environments where evapotranspirative fluxes are really high, okay? Now, another super cool adaptation that has led to a different type of separation of CO2 uptake and CO2 fixation is crassulacean acid metabolism, or CAM photosynthesis. Okay, this is really, it's evolved multiple times, so it's present in 10,000 species across 33 plant families. And a neat thing about this is that photosynthetic rates relate to the ability to store the 4C carbon compound, so this organic acid, crassulacean acid, that is produced. So many CAM plants, so the ability to store this acid in this special vacuole dictates the photosynthetic rates in these plants, and so the better your storage capacity, the higher your photosynthetic rates. And so what has happened is that many of these species that use CAM photosynthesis are also succulent plants. So we tend to have sort of succulent desert species as plants that are most commonly use CAM photosynthesis. Okay, so and what happens here is that there is temporal separation of CO2 uptake and CO2 fixation. So in the nighttime, the stomates are open. This is the coolest, most humid point in the day. This is when we'll see the least water loss. And so this is really about minimizing water loss and improving water use efficiency. Okay, so CO2 is taken up through the stomates at night. And then during the day, the stomates are closed to prevent water loss. That CO2 comes into the mesophyll cell at night and is converted into crassulacean acid. So this C4 organic acid using PEP carboxylase. Okay. All right. So that is what happens at night. And during the night also, we have starch conversion to a C3 compound that forms the C3 compound that is combined with CO2 using PEP carboxylase to form this organic acid. Okay, so we have breaking apart of starches to produce these C3 compounds to feed this reaction. Now, during the day, the stomates are closed, so no more CO2 uptake during the day, but this is when photosynthesis happens because, of course, we need light to fuel those photosynthetic processes, okay? And so during the day, these C4 organic acids are released and release CO2 to the Calvin cycle. And so we have a C3 compound that's released and converted back to starch, which will then be reconverted to this C3 compound during the night. That CO2 is can't escape through the stomates and is instead used in the Calvin cycle. So another carbon dioxide concentrating mechanism that helps the plant to avoid water loss. And the idea is that water availability is really a driving force behind the evolution of CAM. So we see CAM plants, so as I mentioned, a lot of CAM plants are succulents, but we also see CAM photosynthesis used in epiphytic species like these orchids we see here. And epiphytic species, they grow and are rooted in little thin humic layers in the branches of tropical trees, way up in the canopy. So these are environments that are not necessarily exceptionally hot, not hot like desert hot, but are very water limited. And we also, so that's one line of evidence that suggests that CAM evolution was in response to water limitation. But also the fact that we see this ability for some species to be facultative in their use of CAM photosynthesis. So when it's dry and they need to really conserve water, they can operate as CAM, the CAM pathway. When there's sufficient moisture, they can shift back to a C3 photosynthetic pathway. So their structure, unlike C4 plants, is pretty much the same as a C3 plant. They just use this temporal, they shift to this CAM photosynthesis and have these additional vacuoles to store these organic acids. So the idea is that CAM photosynthesis, it really evolved in response to water limitation. And we see this very commonly in many dry environments. Okay, so just to sum this up, and this is our last slide, but here are different types of photosynthesis. So our traditional photosynthesis, C3 photosynthesis, where CO2 comes in and is fixed by the Calvin cycle and sugars are produced. C4 photosynthesis, CO2 is taken up and then converted to a 4-carbon compound, shifted into these bundled sheath cells where it's released again, So there's this CO2 concentrating mechanism. And so again, using the Calvin cycle, that CO2 is fixed and produces sugars, but far fewer mistakes with respect to photorespiration. And then finally, we have CAM photosynthesis, where we have CO2 uptake at night, conversion to this 4-carbon organic acid. And then during the daytime, the carbon dioxide is released from that 4-carbon organic acid and fixed in the Calvin cycle as per C3 photosynthesis. So these are this is sort of the amazing diversity of photosynthetic pathways that have evolved in response to different environmental stressors and support primary productivity across the planet. And with that, I will say goodbye. Have a great day, and we'll see you tomorrow during the synchronous lecture.